of the class. So 40% is quite a lot, right?
Yes.
It's up to you. Since there are 3 of us, of our students, what do you think?
I think we should start because I hope that they may join.
Okay. Ming, is Ming here?
ค่ะอาจารย์
เราเรคคอร์ดไว้อยู่เนาะ
ใช่ค่ะได้คอร์ดไว้อยู่ค่ะ
Okay, so okay, จุฑารัตน์กำลังติดต่อให้อยู่เนาะจำชื่อไม่ได้ทีจุฑารัตน์นี่ชื่ออะไรนะออมค่ะออมอลิสาที่อะไรนะ
ปังปอนนะคะอาจารย์
ปอนอลิสาเป็น
จอยค่ะอาจารย์
โทษนะขอโทษเราเจอกันน้อยไปนะโอเคปอนกับจอยโอเค Dr. Now I won't forget your name
My name only short.
So before we start, shall we have a chat? Joy will do the Opistokis, right? The research in Opistokis, right? Joy นี่ไป
Yes. Joy answer อาจารย์
มาแล้ว And Dr. Now still doing
Yes,
the
อาจารย์
cholangiocarcinoma or what?
อาจารย์ actually the committee I have only OP2 case but it a little bit related with cholangiocarcinoma but I'm not study about cholangiocarcinoma. I study OP2 case endemic in Cambodia and the prevalence we use for detect we use PCR and formalin ethyl concentration technique, but I'm not deeper into cholangiocarcinoma, just the associated with cholangiocarcinoma.
Okay. And
patient, yes.
Okay, thank you for changing your name for me. Is that you changing the name for me? I can see your nickname now. Thank you so much. So, and what Bond will do in your research?
My research topic, right?
Yes.
And now not yet.
I mean, what immunology would help you in your project for the students? So I can emphasize on or stress on according to your need.
Okay.
I don't understand at all, but you mean this subject to help my research, right?
Yes. Why do you register for the immunology? Since it is not the absolute requirements, so it's different from the previous semester from the concept. Those are the requirements. But this is not the requirement. So perhaps I can teach or discuss with you for what you need.
I need to know about the immunology for คืออยากจะเรียนรู้กระบวนการออกมานะคะอาจารย์เพื่อแบบ to approve my research.
Okay, I don't understand in English. No, just joking. Okay. So if you don't have any particular interest in the immunology field, I may do the general lecture for you, but in case you have any question, actually I'm not an expert for this topic, but since I have lectured this topic for like 3 years for all of you, so but my slides, I feel that the slide is not very updated. I'm so sorry because I got contact from Ajahn Kanyarat very shortly, so I don't have a time to revise it. It's pretty old, but hopefully I will give you some concept about our immune system and then the overview of it. Okay, but feel free if it is too much, just stop me anytime. And but my exam is as before, as usual, it would be quite difficult. So my reference, I already got the reference. One moment. ครับสวัสดีครับครับส่งไว้ที่สำนักแพทย์ตึกรปภเลยครับขอบคุณนะครับครับ So after our lecture, what I would strongly recommend you is on the orange side, on the pink side. The book is called Cellular and Molecular Immunology by Abbas. Abbas is the author and editor of this textbook. So most of my lecture just come from this only one resources. So you don't need to find, you can find any other immunology textbook, but what I lecture is mainly from this one. And I believe many immunologists would use this as one of the immunology Bible. Okay. ที่พูดเยอะคือประวิงเวลาไว้อะนะก็จะรออีกไหม
Okay.
Okay.
One student from Cambodia, he cannot join Ajahn because now he in the hospital.
Oh, he's in hospital doing service or become a patient himself?
I'm not sure. He just said that he is in a hospital. He cannot join, but
Okay.
he follow the video from record later.
Yes. Okay. So anytime they can go back and do the record.
Yep.
And also, I think I will have an assignment about the journal, some journal article, 1 or 2 journal for all of you to work out, and we'll discuss next week in the hour of the innate immunity next week, next Thursday morning, I think. Okay, shall we go ahead?
Yep.
Right. So what I would give you an overview. I am responsible to so now I'm starting. Okay, I will tell you for the 2 topics. The first is about the overview, but the overview carries loads of contents. It contains like from chapter 1 to chapter 4 in Abbas. Okay, and the second part is the innate immunity, is the smaller part but also as important, and you know what innate immunity is. Okay, so I will give you some overview of the general immunity itself, and then I will go to the structure or organs, cells, tissue, and molecules, some important molecules in the immune systems. And after that, I will focus on the molecules called mediators, chemokines, cytokines, and their receptors. How each immunity talk together. They need some message, right? You can think of mediator, chemokines, or cytokines as the message communicating between cells, right? And these are very important when you study the immunology. Okay, in the next slide. So just to give you some idea that why immunology is very important. I'm the neuroscientist, but as you see, for the discovery in the Nobel Prize in Physiology or Medicine, you have like physiology, biochemistry, genetics, microbiology. Some chemistry can become involved in the medicine, but in the field of immunology, 20% of the people got the Nobel Prize in physiology or medicine, got the Nobel Prize from the work in immunology. So there must be something very important to us, to our human health. And if you count microbiology like tuberculosis or like Helicobacter pylori, who get they somehow combining with the immunology, you can count up to 30% of the immunology related field. In my interest, our great guy only get less than 10% in the Nobel laureates in physiology or medicine got the Nobel Prize. So if you want to become a immunologist by statistics, you get like over twice or triple chance to win the Nobel Prize than me. Okay. And first lecture I gave in our translational medicine course is in 2018, and that also worked about the negative immune regulation for the cancer therapy. Okay, and in the chemistry also they study about the structure in the chemistry. Who study the antibodies, which is a part of the immunology, also got the Nobel Prize in that year. And of course, you cannot avoid it. The COVID era has come, and people are crazy now about the vaccine. And of course, there are a lot of vaccines to protect us from the virus. It's unlike bacteria that we get the antibiotics to kill the bacteria. But in fighting the virus, we need a lot of our immune enhanced mechanisms, and that is vaccine. And the vaccine is developed from the principle of knowing immunology. Whether it is mRNA based or whether it is the dead particles, you need the T cells to function, you need the antibodies to function. Okay, so I begin with this slide because just to emphasize that how important it is that immunology is to the world. Okay, let's start about the general immunity. Okay, in the overview of the immune response, what is immunity? Immunity in the original meaning is come from the word from the Latin language. The Latin language is called immunitas. The immunitas is come from the protection from the legal prosecution or the protection from the taxing เก็บภาษี and this is when the Roman politician get elected into the cabinet or into the administrative office, they don't have to pay tax, so they are protected from the taxation. So we adopt that term, the immunity, from the word immunitas. In the immunology, our immunity has two types of function. One is in physiologic, and when it become it making mistakes, it can cause the disease or the pathologic condition. But what is the normal function of the immunity? We can subdivide it of the normal function of the immunity or the immune response into two types. The first is the original meaning, which means is this defense against the invaders. Dr. Now disappear. Shall we wait? Tick-tock, tick-tock. Is he around? Okay.
He lost.
Okay. The original physiologic meaning is the defense against invaders. The invaders, whether it is the infectious microbes, fungus, bacteria, or parasites, or others, virus, for example. Or in human or animals, you can walk and got some kind of wounds or nails that pinch in your arm or your hands or your feet. That's the infectious, non-infectious foreign substance or foreign material stuck into your body. We also got the immune response to that as well, to avoid further deterioration of our tissue. And now the immunity is not just that, not just the external invaders, but this also, actually it is also against ourself, our normal self or our abnormal self. Our normal self include the dying tissue or damage, dead cells or debris from the cell products. Sometimes we have to degrade that by the process of immunity. And the cell that misbehave is called the cancer cells, for example, or hyperplasia, metaplasia, metaplastic cells. You also have some immunity against those cells as well. But not always that immunity is good to us. When it is doing something wrong to us, it can cause our disorder as well. You may learn it later. The first one is overreacted immunity, like hypersensitivity, แพ้ยาแพ้นู่นแพ้นี่เนาะ or it is against the normal cells. Our normal function is like the autoimmune disease, okay, or in some certain pathologic function, it can become malignant itself, like leukemia or lymphoma. It is itself become the cancer by I should not call the cancer, the oncogenic function. I forgot the term, sorry. มะเร็งนะ okay. So, and the immunity, when we talk about this, is about generated from ourselves, but actually we can get stimulated or get it from somewhere else. So we should know that our immunity may be acquired. Coming later, when you are born, you don't have this kind of specific immunity, but you are acquired or get it later in life. It is called active immunity. Or you can get transfer or confer from outside your body for a short period of time. You call it passive immunity. You don't get the stimulus, you get the whole antibody or protection immune system from outside and transfer into your blood or to your body. that is called passive immunity, and you will study later, of course. This is only the introduction. But so up to this point, ใครนะอุ้มใช่ไหม just join, right? Okay.
อาจารย์สวัสดีค่ะ
Okay, just quick recap that I would like to emphasize that the immunology is important, and a lot of people get the awards like Nobel Prize from the immunology, so it is important. And I explained about the what is immunity is from the Latin word. And usually immunity, we think of defense against invaders, but actually it can have the defense against our normal tissue like dying or tissue debris or the cancer cells. But immunity is not, sometimes it misbehave and it may become like ภูมิแพ้เนาะหรือว่าแพ้นะ hypersensitivity, or it can become the autoimmune, or it can become the cancer itself like lymphoma or leukemia, or it can doing harm to us by rejecting what we transfer it into, like transferring blood or organ transplant. It can rejecting those as well, so that might be the function of immunity that we don't want. And lastly, the immunity can be stimulated or transferred. If it is stimulated, initially we don't have it at birth, but we can stimulate by any means, and we get the period enough. It is called active immunity. But if you get for a short period of time, you don't get self-stimulation, but you get the product of the immunity like antibody or the plasma or what to protect. We call it passive immunity. Okay, we go pretty slowly. Next. Let's start with the classic example of the immunity. We often think of the protective immunity against the microbes, our enemy, long-term enemy. It's so small, but it is dangerous to us. We can divide the immunity into two types. The first one is the innate immunity. It acts very fast. It acts quite early, but it's not very specific. It involves, it can be classified broadly into two types. One is the physical-chemical immunity, like what? Like our skin, like our mucosa is already like physical protective barriers from the invaders. And it also involves, we will talk much about the cellular defense, like the original cells that help to expel the invaders like microbes. It has the limited diversity or specificity. Usually this innate immunity, the majority of the innate immunity, if excluding the physical chemical property of the innate immunity, in terms of cells, most of the innate immunity are phagocytes, the cells that can eat up the invaders. These are, most of them are innate immunity. For the proteins or the molecules like that help a lot in innate immunity, there are a lot of proteins or molecules which I will explain to you in the lecture of the innate immunity. But one of them you should know is the complements, and this word comes to you again, and I explain to you later. Contrasting to the innate immunity, the most powerful the more powerful of the two immunity, we call it is adaptive immunity. It is very diverse, and it is so diverse that they can rearrange the structure or the patterns of the immune response to almost any kind, like I don't know, in millions of ways to act against the invaders. And when it knows what exactly the enemy is, it is so specific to that invaders, to that enemy. And the good of the immunity, this kind of immunity, is they have memory. So the vaccine, the vaccines that we develop, we don't have this property of the adaptive memory, making a lot, making very specific, and remember when it attack or when you get infected, and don't harm to your body. So it means that they can discriminate self from non-self. Most of the cells in the adaptive immunities are lymphocytes, as you know. But the innate immunity and the adaptive immunity, they don't work separately. They work together. Once you get the invaders come to the innate immunity, there must be some way of the innate immunity to communicate and transfer the response to be more specific, more memorable, if it is important enough. So we need the communication of the two. So the innate immunity have some specialized cells that communicate and signal the lymphocytes to act very specific. These are depending on the type of the antigen. Do you know the word antigen or not? Any part of the invaders or any part of the foreign things or anything that can stimulate the immune response that come in. You need the special cells to present. Some of the innate immunity cells are phagocytes. They can eat a lot, but some has to talk to the lymphocytes to convert this signal of recognizing invaders, and our body can respond to the invader more specifically. This molecule to get the communicate. These cells, we call it antigen presenting cells. Okay. And after innate immunity has the antigens, and we get the antigen presenting cell present antigen to the lymphocytes. So the lymphocytes can recognize and proliferate and differentiate, and it becomes these lymphocytes become the memory cells or the effector cells to signal and destroy our invaders. In this case, most of the lymphocytes involved in our immunity, we call it the B cell and the T cells. Okay, or lymphocyte ชนิด B กับชนิด T เนาะรู้จักอยู่แล้วใช่ไหม You already know this kind of these two, right? There are some multimedia explaining about this process. You can look at it in the YouTube. Next overview is the humoral immunity. The adaptive immunity. The adaptive immunity can be divided into two types. One is called the humoral. Humor in this case means fluid. Okay. Humoral immunity, and the second one is less involved in the fluid. Is more involved with the cell-mediated immunity, HI and CMI. The humoral immunity, the primary cell responsible for producing humoral immunity is called B lymphocytes or B cells. and the product dissolving in the fluid, in the plasma, in the serum, in the lymphatics, floating around. We call of the this product of the B cell are antibodies. And this is the major defense against the extracellular agents. And when the antibody acts, they can signal to many pathways, but part of them they can do enhance the process of the phagocytes to eat the invaders, or they can act together with the protein called complements to enhance the lysis function or phagocytic function, or the inflammation to occur. Okay. As for the cell-mediated immunity, most of them are the function of the T lymphocytes. And what these T lymphocytes do, they secrete a lot of signaling molecules. And one of them we can divide it, the T lymphocytes into broadly divided into two subtypes. There are a lot of T lymphocyte subtypes you need to know, but what you definitely need to know that T lymphocyte, there are at least two types called CD4 helper T. CD4 what CD4 helper T cells do is that it secrete a lot of cytokines and also help the macrophage ingestion and enhance the B cell antibody production. The other type of the T cells, we call it CD8 positive cytotoxic T cells. They don't actually kill the invaders, but they tell the infected cells to die, like holding the gun that, oh, you get infected, kill yourself. So the infected cells will have to undergo self-suicide, which we call in the cellular level, we call it the process of the apoptosis. T cell also has CD4 positive cells, which mean the marker on the surface of the T cell contains the CD4 molecules that has other functions like regulatory function, tell the signal when the immunity should go on or when the immunity should stop, for example, or when this immunity should be memorized, for example. It's the role of CD4 positive T regulatory T cells. There are a lot of T cells, but in your level, I think definitely you need to know there are two types, CD4 plus and CD8 plus. Just to let you emphasize about the cardinal features of the adaptive immunity that is different from the innate immunity. This is just the summary that adaptive immunity must be usually has more specificity, diversity, memory, and they can expand or self-proliferated the same type of the antibody producing cells or same type of T cells with a lot of cluster. We call it clonal expansion, and has a specialized function in the managing invaders like specialization, specialized differentiation, and it help work together with other proteins to help with the blood clotting, the contraction, or the homeostasis. And the good immunity should be able to recognize self. They don't destroy themselves. There are some exceptions, but this is the general rules of the adaptive immunity. The molecules of the playing a lot of part in this communication between immune cells there are a lot of chemicals involved in, but the first one you should know, we call it the cytokines. The cytokine, the definition of the cytokines, there are a lot of cytokines, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, interleukins or tumor necrotic factors, interferons. These are molecules, we call it cytokines. These are secreted proteins mediated and regulated both innate and adaptive immunity. For the chemokines are subsets of the cytokines. Cyto means cell, kines mean secreted molecules. Okay, the chemokines are subsets of cytokines, but they mainly attracting or drawing movement of the immune cell together. Okay. Example, there are a lot of cytokines as I mentioned, but after finish this class, you should know these three pretty well. The first one is tumor necrosis factors or TNF. The other one is the interferons or INF, and the interleukins. Interferon is the IFN, not the interferon. Sorry, I mistyping. Interferons. These are standards abbreviation, and there are a lot of interleukins communicating between lymphocytes and macrophage. Okay, so that's the overview. I give the definition of the immunity. I give the definition of the innate immunity, and I give the definition of the adaptive immunities. And the adaptive immunity has the specialized property like diversity, specialization, memory. These are hallmarks. And the representative cells in the innate immunities are phagocytes. The specialized, most of the cells in the adaptive immunities are lymphocytes, and lymphocytes can be divided into B and T. B cells are humoral, produce antibody dissolving in the fluid. For the T cells, they are cells that are signaling to other cells, and they secrete the cytokine, use cytokines as the signaling molecules. And we go further a little bit to know about the terms of cytokines, and there are subsets of cytokines. Up to this point, do you have any questions for me or what? Or not? Shall we go ahead?
Yep, Ajahn.
Okay.
Go ahead. Yes.
Next, that's the overview that what immunity do. And now we know a little bit about the anatomy or the type of cells and the tissue. Okay. The immune cells, they are, you can call it the immune cells. They are almost everywhere in many tissue. They can deposit in lungs. They can deposit it in our oral cavity. We have the tonsils. At the arm we have a lymph node. There are like Rọpọpọ, it's like the security guards. It resides almost everywhere in our body, but to be able to communicate, they need the connection. The quickest way of the immune cells to act, they need circulation. We already know that our circulation, mostly we travel in blood, but we also, in immunity, we have a specialized system of the circulation as well. They are called lymphatics. ทางเดินน้ำเหลืองนะ the travel passage for the immune cells and cytokines, and the passage of whether these immune cells travel through the blood or to the lymphatics, we must have the checkpoint or the security guards in specific part of our organs, like lymph node, for example. We call it sentinel in the group of the immune cells that stay there in the tissue in checkpoints. The most effective immunities, as I would like to emphasize, the adaptive immune cells, because they have an ability to learn and memorize, and ability to communicate with other cells pretty quickly, and they have what I would like to say, I forgot. The good of the way, apart from having the blood circulation, the lymphatics is another way of the immune cells to have efficient delivery of the immune cells in particular, and especially when they become specialized immune. Okay. Before we go into the tissue, you should know some certain properties. There are a lot of immune cell types, and these are examples of the main effector immune cells that you must know. Definitely, the immune cells that you must know includes the phagocytes, and the phagocytes, any cell that can eat. There are a lot of cells that can eat invaders, but only two that you need to remember. Some cells can eat, but they're not doing the main function as eating. But these are the cells that do the main function of eating. They are macrophage and neutrophils. I think you can recall it from the secondary school, what macrophage is and then what neutrophil is. Right? And other immune cells that travels through the blood or involved in the certain tissue that you can get allergy, we call the mast cells, basophils, and eosinophils. Okay, I don't emphasize much about these cells. You can look up later in the detail if you want to. The other part is the linkage between the innate immunity with the adaptive immunity. We call antigen presenting cells. They present antigen to the specific immune cells to get the specific and expand the immune response. The most important antigen presenting cells you need to know is the dendritic cells, and I will tell you later what dendritic cells are. There are some cells that look like the lymphocytes, but actually they are not, but still doing some immune function. We can call it lymphoid cells. Again, this is not the emphasis. The next one, you need to know a little bit further about the B cells and T cells. What do they do? Which I already tell you. Later on in this part, in this chapter, you need to know that the immune systems, they can be divided into two types of the immune tissues. The first type is the primary immune tissue. The first site that generate B cells and T cells, we call it, and the hematopoietic stem cell, white blood cells from the bone, call it bone marrow. And another primary immune tissue is called thymus. For the secondary immune sites, and the passage of the immune system apart from the blood, called lymphatics, lymph nodes, spleen, and regional immune system. Let's talk about the cells of the immune system first. Okay. We have a lot of red blood cells in body. Is contain terms of like millions of cells per microliter, but for the white blood cells, which doing the regular immune response, we got usually no more than 10,000 cells per microliter, and 60% of these are neutrophils, and we have like 20 or more percent, 20 to 40% are lymphocytes. Monocytes, I'll tell you later, it will become tissue macrophage later. Give you an idea what each cell look like in the cartoon. Okay, and next, we should know the first cell types which regulate mostly in the innate immune system. Is called the phagocytes. The phagocytes, most the cells that can eat other invaders. The phagocytes can be divided into two types. One is from the circulating white blood cells, the neutrophils. One is usually reside in the tissue, or it can convert it from the monocytes in the blood to the cells that can eat. We call it macrophages. They do function as they can identify, ingest, and destroy microbes. Okay, neutrophil I already talked is the circulating white blood cells from the originally they proliferated and differentiated from bone marrow. For the macrophage, you should know that the macrophage can, after birth, there are some macrophages that already stay in each type of tissue in the lung, in the liver, or in anywhere. We call it resident or tissue macrophages. And we have another type of macrophages that are differentiated from the circulating leukocytes, or from the circulating, more specific is the monocytes. Macrophages can get from two sources. Actually, I emphasize about the macrophage of the phagocytes. They contains about neutrophils and macrophage, but actually there are another type of cells that usually resides in the tissue that can eat up, but they are not good at eating. They can only eat the digested part of the invader, and after once they digest, they present that part to other lymphocytes. These cells that can eat but not good at eating, but good at presenting, and has a lot of arms. Dendrite mean tree-like structure. We call this, and this they present by, because they have a lot of hand, just like cartoon, right? If you have a lot of hand, you can reach out and present a lot of things to other immune cells. Okay, these cells are dendritic cells. They are not good at presenting antigen. Dendritic cells is a subset of phagocytes as well, and it is also part of the antigen presenting cells. And I will give you more strict definition of what antigen presenting cells are. And these phagocytes, once they engulf or exocytosis or endocytosis, engulf the invaders into the cells, they can kill the organisms by doing what? The first one is by reactive oxygen species, by creating oxidative stress And the oxidative stress can divide it into two types. One is by the pathway of the nitric oxide, and the other one is by reactive oxygen species. The other way of killing the invader is doing the enzymatic process. They do use the lysozyme as the digestion. Okay, to help you remember what they can do, I give like some Thai words as well, but for the English, there are some definition as well. Okay, the neutrophil, what they can do, they are very aggressive. Okay, very aggressive. They want to eat everything. They want to eat everything. Okay, and they are very aggressive. Okay, the macrophage is always hungry, but not very aggressive. Less aggressive. Okay, and they has some degree of the antigen presenting capacity better than the neutrophil. Okay, the ones they have very small mouths. They like to chat rather than eat, and they like to give things to others. Yeah. These are dendritic cells. Okay, they are very lean comparing to the macrophage or neutrophils. Okay. These are examples of the antigen presenting cells. I don't think I have time to go into detail, but perhaps you need to remember to read it, and these are tables that for you to go back and review it. And if you get a problem with understanding, you can get back to me anytime. Okay. Okay. Just to explain you more a little bit about the origin. Actually, the origin of the dendritic cell is pretty unclear, but you can think of the dendritic cell similar to the macrophage as well. Let me example of the macrophage. Macrophage, if you look at the lower picture, that the macrophage can, early in life, the macrophage come from the same series of the hematopoietic stem cells, but they travel to deposit into the destination, the tissue destination, such as kidney, spleen, liver, lung, brain, very early in life, and they proliferate and differentiated within that organ. When you grow up, they still differentiated and proliferated within that organ. They don't need to travel. They just reside in that organ. We call it is like tissue macrophage. Sometimes it has specialized name. Resident macrophage or tissue macrophage has specialized name. This macrophage, if it is resided in spleen, we call it sinusoidal macrophage. If it is inside the lung, we call it alveolar macrophage. If it is resided in the liver, we call it Kupffer cells. If it is resided in the brain, originally from birth, we call it microglial cells. But the macrophage can come from the newer version when they got activated or get the process of inflammation by the circulating, by differentiating from the blood cells. That blood cells that when get enough signals, they can turn into the macrophage. These cells are called monocytes, and these monocytes has the origin come from the bone marrow when you get old. and the turnover is pretty quicker. Okay. And what the macrophage can do, perhaps you can learn it a little bit, that macrophages from the basic science in translational medicine that we have in the part of the inflammation and the healing. So the macrophage can do broadly, we can classify the function of macrophage as 3 functions. The first one is eating. Eating, we call it the classical macrophage, by killing microbes by ingesting. It can also ingest dead host cells. It can also participate in inflammatory process. Actually, from the acute inflammation and chronic inflammation. But the macrophage can do the remodeling, repair, and the scar formation as well. We call this macrophage as the alternative macrophage. And the macrophage, as I told you, they can talk with the T cell. They can be acting like the antigen presenting cell itself as well. Okay, we talked too much about the macrophage of the phagocytes, and the neutrophil doing similar thing, and you can go back and review that table again. For the mast cells and basophils and eosinophils, usually it's circulated in the blood. When in circulation, the mast cell, basophil, eosinophils share the same similar look because they contain granules, but different in colors. Eosinophil is often stained red with the hematoxylin in eosin. Eosinophil acts a lot in the allergy parasites, in response to allergy parasites and foreign bodies. Basophil acts more to the allergies, but the main function of the basophil we still don't fully understand yet. But if this granule immune cells resides in the tissue, we think that it might become from the eosinophil. But if it is resided in the tissue, we call them mast cells, doing the similar function to the basophil and eosinophil. And they can contain the certain antibody called the IgE and IgG. Okay, let's have a look at the strict definition of the antigen presenting cells. Actually, antigen presenting cells, you know the function, it's kind of sending the message from the non-specific, less specific immune response to the more specific, more memory, more diverse, more proliferation to the adaptive immune response. We can classify it into the antigen presenting cell that triggers the adaptive immune response into 2 types. One is the professional antigen presenting cells. They are born to present the antigen. They are born to present the antigen because they have special protein เกิดมาเพื่อส่งของนะ like Grab, like Food Panda, like Postman นะ like 7-Eleven delivery they are born to send the signal to the adaptive immune. They have the same kind of motorcycle, presenting signal molecules called major histocompatibility complex or MHC class 2. This motorcycle, this MHC is very specialized to trigger the immune system. They can carry antigen a lot. So because this cell has specialized delivery tools, so they are professional. The delivery tools means delivery proteins to present antigen. It is the MHC class 2. The cells that contain MHC class 2, for example, the example of the cells that contain MHC class 2 are dendritic cells, macrophage, B cells, follicular dendritic cells. These are professional. They are born to be to present antigen to the adaptive cell immune cells. พระเอกของเรื่องนี้นะ the most important one is the dendritic cells. The second type of antigen presenting cell, they can deliver if they pass by T cell นะ by accident, by if they sick too much นะ these cells if they sick too much or they are broken นะ they are broken, usually they have like a basket of and presenting basket. These are all nucleated cells. Nucleated cell in our body, skin epithelium, neuron, อะไรนะ cardiac myocyte, skeletal myocyte, cartilage, fibroblast, any kind of cells that contain nucleus. If they are sick enough and when they are about to broken, before they are dying, they just say to the adaptive immune cells that, hey, we are too sick, immune cells doing something like that. So they present another type of, they can act as antigen presenting cells by presenting specialized molecule called MHC class 1. So all nucleated cells can be antigen presenting cells. For example, if they are for a certain condition, for example, like when they are sick, we can present the antigen that so the immune system can know that, oh, we are sick now, we should do something. Do you think red blood cell is antigen presenting cell or not? If it is it professional antigen presenting cells or non-professional presenting cell, antigen presenting cells? Anyone?
Red blood cell อาจารย์ Red blood cell is not a kind of antigen presenting cell because it's not a function to kill any antigen, but white blood cell is the group of presenting antigen presenting cell.
You think
It's
it is
what I understand. I think
so
red
you
blood
think
cell
it's non-professional?
Red blood cell non-professional.
Okay, it's incorrect. And just for example, because red blood cell does not contain nucleus. Red blood cell
Yeah.
has no
Okay.
nucleus, so
Yeah.
they cannot be antigen presenting cells. Okay,
Yep.
so that is a quiz to keep you in mind. Okay. So when we talk about the antigen presenting cell, you need to know a little bit further about the dendritic cells. The dendritic cells, usually the main function of the dendritic cells present antigen to T cells. They can have a lot of specialized function, and sometimes they have specialized shape. If the shape of the dendritic cells look pretty big and I forgot what to say, but and look like the plasma cells. Plasma cell is like the B cell that produce antibody. Anyway, look different type of another type of dendritic cells. They can produce, we call it plasmacytoid dendritic cell. These cells can produce interferon alpha. You learn it later. It's another type of cytokines specifically reacted with the viral immune response. Immune response to virus. If it is the dendritic cells, certain dendritic cells are specialized in the inflammation that produce produced in the certain type of inflammatory process. We call it is the inflammatory dendritic cells. These inflammatory dendritic cell has a specificity. Usually dendritic cells stimulate T cell, okay, but inflammatory dendritic cells stimulate specific T cell type. We call it T helper 17 cell. Just for example, that dendritic cells can be classified into several types. If the dendritic cells mostly resided in the lymph node, sometimes it helps with the T cell to stimulate B cell function. We call it, and usually it functions in the lymph node. We call this type of dendritic cell as follicular dendritic cells. So dendritic cell does not present antigen to any T cell. Sometimes they have specific function to specialized T cell as well. Okay, what is CD? That is dendritic cell DC. CD, if you remember, I need to recall to define it a little bit more. In some previous slide about the T cell, T cell we have CD4 กับ CD8 and CD8. What is CD? There are a lot of type of CD. CD actually means the full meaning of it. It is cluster of differentiation. The cluster of differentiation is broadly classified is any surface molecules, any molecule that expresses on the surface of the immune cells, of the cells. No, sorry. So not I forgot what to say. Actually, it's not the any surface molecules that marked on all type of cell. Sorry, surface molecule that mark on all type of cell, but they can be classified into different CD by if you put different antibody and you can differentiate group of cell into cluster. If you put antibody to cluster of differentiation 4, you get the T cell helper, the helper T cell, for example. But actually, the CD is the surface marker on any type of cell. งงไหมเข้าใจไหม
อาจารย์อธิบายภาษาไทยเพิ่มได้ไหมคะ
It's not fair for Dr. Now if I translate it into Thai. So I use translated into Thai and then I use translated into Cambodian, right? No, I cannot. CD is the cluster of differentiation. It's the any surface cell surface molecule. You have the cell membrane, and you sometimes you have protein, you have glycoprotein on the cell membrane. But that glycoprotein or protein that on the cell membrane, they have certain type that T cell, this T cell number 1, T cell number 2, T cell number 3, T cell number 4 share the same surface molecule. And if this surface molecule, one of many, one of this surface molecule. If you put one antibody to be in, and you can differentiate whether T cell number 1, T cell number 2, T cell number 3 are the same group, but T cell number 4 is not different. Yeah. So we call this in the same group, T cell number 1, T cell number 2, T cell number 3 are the same group of cell or the same cluster of cell. That protein which is marked by the same antibody, we call they share the same cluster of differentiation. Yeah. Let's say this antibody that group this T cell are called the antibody to CD4. It means that this T cell number 1, T cell number 2, T cell number 3 are CD4 positive T cell. These are T helper cell, but not the T cell number 4. No? T cell number 4, they are not tagged. They must be other CD. You use different antibody to tag in. So now you know that if it is tagged by specialized antibody, that is CD4 cell, all of them are T helper cell. If they are tagged by CD8, they are cytotoxic T cell. If you tag the certain antibody with CD134, for example, I guess it means the CD134, these are stem cell. They sometimes these surface molecules, sometimes they have specialized function, sometimes they cannot. Just the way you sort the cell out to cluster them.โอเคไหมเอาเป็นอะไรเดียวif you want to sort the cell with the mustache หนวด the cell which is sorted by the one who has mustache and the one who has not, the one who has mustache, that are the same CD, the same cluster of differentiation. But in immunology, they are numbered out from CD1, CD2, CD3, CD4, CD5, whatever, CD100s. Okay. Actually, they are classified a lot of cells. It's not just the immune, but I put it here because later on you have to learn a lot of CD in the immunology subjects. Okay, next type of cell, we talk about the phagocyte. We emphasize on the macrophage and comparison with the neutrophil. We introduce the other type of granulocyte like mast cells, tissue granulocyte, and the circulating macrocyte, granulocyte like eosinophil, basophil. I have give the definition of the antigen presenting cell, and the most important ones are the dendritic cells, as you know. The next type of cells in immunology, which is important, most important, they can do some function in innate immune response as well, but mostly they help in the adaptive function, immunity function. We call them lymphocytes. Lymph แปลว่าน้ำเหลืองเนาะ It's like the liquid fluid. The cells that you can derive from the yellow part of the lymphatic system or the blood. The lymphocytes can be broadly classified into B cells, which is marked by the CD19 and CD21. And the T cells, this is marked by the CD3 positive cells. For the B cells, they function mostly for the antibody or immunoglobulin production. And the B cell, if it becomes activated, it gets larger, like in the picture below. The cells expand in size, and you see the clear the halo, the clear area just in front of the nucleus, and the nucleus is less condensed. It means that it looks in the electron microscope like the clock, หน้าปัดนาฬิกา the clock shape, because it is very active and it is producing antibody. The B cell that is producing antibody, one B cell produce one type of antibody, and this activated B cell, we call it plasma cell or effector B. And you have some, if it is not active in antibody production, some B cell can memorize what antibody they would produce. We call these cells memory B. For the T cell, actually there are a lot more, but the cells that you have to know are CD4 positive helper cell. It plays a lot. If you don't recall anything, at least you must know that in HIV positive patient, when we look how severe the HIV positive or the AIDS patient, there are two parameters that we look at. The first one from the PCR is called the viral load. How much or how many viral that we have in that patient, right? The other factor is how many CD4 that the patient have left. The CD4 T cell in that case means the CD4 mostly for the CD4 helper cell, helper T cell, because the HIV or AIDS patient, if they get without any medication, they will destroy CD4 T cell a lot. So I explain ชักแม่น้ำทั้งห้า from the many ways of the sea, just to let you know what CD4 is. But CD4 T cell can be regulatory in cytokine secretion. Also, if you get the T cells CD3 plus with the CD8, these cells are good at telling other cells to kill itself by apoptosis. We call CD8 as cytotoxic T cell. And again, this CD4 have small population, and this CD8 has small population to keep the memory in our body. We call it CD4 memory T or CD8 memory T. And this memory B or memory T, they can stay inside for years in our body. What I want to tell you, I think too much information by this, but perhaps it's important just to let you know later on. Then, okay. Go to read by yourself then. I want to go into detail now. And if you don't do the cluster of differentiation, how do you differentiate it? Lymphocytes by microscopy, they look the same, but actually the B cell and T cell, they are different in where it come from, For the B lymphocyte, actually initially at the beginning, the lymphocytes come from the same hematopoietic stem cell. In the baby, in the fetus, most of all of the lymphocytes come from the bone marrow. But once they grow up, and if they go into the lymph node, or if they stay in the bone marrow, most of them are differentiated into the B lymphocyte. Certain population of the lymphocytes that during the baby, if they don't stay in the bone marrow, they travel to another organ. It's called the thymus, and got the maturation in that cell. Got the maturation in the thymus gland. We call it T cell. T cell. In the origin, if you look with the naked eye, you cannot tell whether it is B cell or T cell, unless it is differentiated. Like B cell produce antibody, become plasma cell. Certain T cell can become get bigger or change shape somehow. Okay, just to this picture, just to let you know that originally B and T come from bone marrow, but the T cells has to mature in another organ called thymus before it get circulated in the blood. Okay, and by morphology, you cannot tell the difference between T cells or B cell apart from the origin of it. Lymphatic activation. Maybe I don't want to talk about this. Maybe later on. Okay. Are you tired? Yes or no?
Okay, Ajahn, that's okay.
That's okay, but I'm tired. I want to take a break. How about 10 minutes
Yes.
break or 15? 10?
Okay, Ajahn.
10 minutes break. We come like 15:35. Okay.
Yes. Okay.
Yes.
I'm waiting for Pon and Oh, okay, good. Good to see you. So we talked a lot about the cells of the immune system. So now, why do the immune system has to form together, to live together, and they have to organize? They also need circulation, as I told you, right? Because it cannot do the standalone. Otherwise, it cannot. Sometimes it cannot respond systemically enough, rapid enough. So this immune system has a specialized system to permit rapid delivery of the immune cells to the site of infection and provide the response more effectively. So actually, there are a lot of organs or tissues in our immune systems. The first one is called the primary immune organs, the origin where the immune cell start off. We have two organs, one that which generated B cells and all hematopoietic stem cells. The first one is called bone marrow, which resided in the soft and stayed inside part. Part of them are lipids and fat, and part of them are hematopoietic stem cells and immune cells inside the bone. The second, the next tissue for the generative lymphoid organs or the primary lymphoid organs. The second one is in the thymus. Where is in the thymus? Usually it's just like the lipid. It's the yellowish tissue, look like fatty tissue, but actually it's the lymphoid tissue organs above the heart and between the lungs. We call it the thymus glands. The rest of the lymphatic or lymphoid tissues, secondary lymphatic organs or peripheral lymphatic organs. Lymph nodes usually stay in the junction of the arm, like in the armpit or the axilla, in the groin or inguinal nodes between the legs and your pelvis. The deep lymph node inside just lay next to the major blood vessels like aorta or superior or the vena cava, and the lymph nodes in the cervical nodes. And they are interconnected by the lymphatic ducts or ductules. The other organs in the lymphoid, secondary lymphoid organs are spleen. Spleen doing the only not the blood. Wait for a moment. I need to make an urgent call. Sorry. Wait for a moment. 5 minutes.ครับอาจารย์จริงๆถ้าอาจารย์มีข้อแนะนำไหมครับถ้าผมคิดได้ตอนนี้คือถ้าไม่พอแต่ว่าเกินครึ่งนะครับ อาจารย์ก็แบ่งรอบสอบนะครับเช้ากับเย็นแล้วก็กักตัวนักศึกษารอบเช้าเอาไว้ฮัลโหลครับฮัลโหลเสียงขาดหายครับ classes.
Yes.
I do apologize. And,
Yes, Ajahn, that's okay.
okay, let's start for now. To keep it short, I mean the lymphatic organs. The lymphatic system contains the lymphatic organs and lymphatic circulation, like the lymphatic duct or ductus. And for the lymphatic organs, they divided into the generative organs such as bone marrow and thymus. For the peripheral organs, mostly we contain the lymph nodes, spleen, and the regional immune system. Like if it is the similarly to the lymph node, but it does not contain capsule. It contains the mucosa cover. If it is in the intestine, we call it the gout or gut associated lymphatic tissue. And if it is covered with skin, we call it the cutaneous associated lymphatic tissue. And if it is within the mucosa or in the oral cavity, we call it the mouth mucosal associated lymphatic tissue. Okay, let's talk about the bone marrow. The bone marrow is if anyone who has the experience studying the embryology, most of the immune cells is derived from the blood cells precursor, and most all of the blood cells initially it is produced in the yolk sac, and then it is then produced in the liver and spleen, and then in the fetus just before birth it moves into, most of them it is born in the bone marrow. For the immune system, we count from the bone marrow. If it is stay from the bone marrow and then it mature and go into the lymph node and spleen, these are B cells. But if these immune cells initially from the bone marrow, but then they produce their children and grow in another tissue before going to the destiny in the lymph node and spleen. That organ is called the thymus, so we call this cell T cell. Okay, I think we should go a little bit faster. And what is the importance of the thymus organ? The B cell produce antibody, as you know, and usually the antibody is depends on the learning from the cytokines and the antigen presenting cells to learn to signal what clones of the B cell that they should pick up. But the thymus, it is a little, the T cell, it is a little bit different. They need to learn. The T cell, the most important thing. So the B cell is when in order to know which antibody we should produce, the B cell will be only one clone of B cell will be selected out and produce that special type of antibody to respond to the disease. But the T cell is different. Originally, there are a lot of T cell, a lot of T cell type that recognize many antigen. So if they recognize everything, eventually these T cell, if we allow them to grow out, to go up, some T cell will destroy our normal tissue organ. So this T cell must go into the school. So the thymus cell has a specialized another type of cell is called the thymic medullary epithelial cell or medullary thymic epithelial cell or TMEC or ETMC or MTEC. This is like a normal cell that present normal antigen, normal cell antigen. Any T cell that bind to the normal to the normal body antigen, you die. So in this case, the only T cell that are left are T cell that don't recognize self as a foreigner. Only recognize the invaders. So this T cell, this process, it means that the T cell need maturation in the thymus organ. Okay, thymus, and before it becomes the mature T cell. But they need to learn again later which specific T cell they should produce more in later life when they got infected. Okay, that's the function of the T cell. I forgot. We have some animation. I already forget. The lymph node. The lymph node is they are located many places. Usually they are the junction before getting into the central, very important part of our body. Lymph node, or the way the lymph node can activate it is the sentinel, ป้อมยาม. They collect antigen from the lymph or the lymph pathway. When the lymph node collect the antigen from the any portal, any doors of entry, the lymph node can stimulate the adaptive immune response by itself later. This lymph node, in the middle, if this in the blue, this is the capsule, and this the follicular. The follicular lymph node follicle contain a lot of B cells. If as you get inside, T cells stay inside. It's called the T cell zone or medullary zone. Lymph node. Okay. When the antigen get in from the portal entry, lymphatic portal entry, the naive lymphocytes from the blood vessel, whether it is artery or veins, gets in, and then they mix and stimulate that immune response, and then they go out again to the efferent in the outway of the lymphatic vessel, and then recirculate it into the blood. Just here and let it pass. It's not. I'm not going to emphasize much about the pathway, but just want to let you know that the lymph node is the place where the antigen can stimulate the lymphocytes to be active as the adaptive immune response, and that's all. The next organ is spleen. Spleen, actually, the organ behind our stomach is called the spleen or Mast. Contain two types of tissue. One is called the red pulp. The other one is called the white pulp. The red pulp usually is the sanctuary susan for the old or the aged red blood cells. I'm not going to emphasize in this lecture. But the white pulp contains the germinal center for the immune cells, both B cell and T cell. We divided into if it is the place where B cell proliferate, we call it B cell zone or lymphoid follicle. Next to the lymphoid follicle, we often found another zone called the T cell zone or periarteriolar lymphoid zone. Okay. The white pulp also help to get rid of the immune products or the any cell part that is digested with the after the infection, and the immune cells act together. And it is also the another important function. I already emphasized the importance of the dendritic cells. The dendritic cells also need maturation in the white pouch as well. Okay. Glucosamic migration. Okay. At this step, one moment. Okay, not too much. I think I will stop with the general introduction, and then I find another lecture to catch you up later. Okay. In order to move or mobilize the leukocytes, this process is called the leukocyte migration. We need we need specialized cytokines. The cytokines that move the red blood cell, or the cytokines that move the white blood cells or the leukocytes, we call it the chemokines. This picture is to show that when we get the inflammation that at the injury site, how the injury site can know to attract more the immune cell to be into the injury site. We call that process the leukocyte recruitment. Somehow the invaders and the localized tissue macrophage must send signal to recruit the leukocytes by sending some cytokines. More specifically, is called the chemokines. And then it draw also other cytokines to secrete that, or the plasma proteins. Another step is called the plasma protein recruitment. And then you can record it from the lecture of the basic science in the concept of the translational basic science in the translational medicine, that it is not just the injured cells, tissue macrophage to act as the inflammatory process. We have the vascular process as well, like the vascular process. We call it the endothelial activation. Endothelial activation, they still secrete a lot of molecules to attract more, to recruit more the leukocytes to roll and stick more into the blood vessel wall and easily to pass through the tissue at the injury site. This is the broad picture, just to review. It's not the what I want to emphasize. And in order to have the immune cells from the circulation get into the injury site, this is the multi-step. From the usually the white blood cells travel in the midstream blood flow, but in some case, if we get the injury on that side, endothelium and at that side express specialized molecules, for example, the selectin, to move the white blood cell in the middle of the blood vessels from the middle to get into the near to the wall. And then they have express a lot of the proteins that attract white blood cells to roll and migrate and transmigrate into the injury site. So it depends on each textbook. In Abbas, they divided into 4 stages, like rolling, activation of the chemokines. This is the vascular process. Stable adhesion, and transmigration to the final tissue. This vascular process needs expressing molecules from the vascular endothelial cells and the expressing molecules within the white blood cell. This family of molecules is not just one name. There are a lot of them. The family of these molecules, we call it together as selectins, integrins. And that's all. This is just to give you the picture. I don't want you to remember of this in the later slides. For this is the subset of the cytokines that draw or migrate the leukocyte. We call it chemokines. The chemokines, in order to act at the white blood cell, they need the receptor. Chemokines and chemokine receptors. Chemokines are here. So, okay, we have the definition. Chemokine is a subset of cytokine which stimulate leukocyte movement and regulate migration from the blood to tissue. And the chemokine can be classified into more general terms, like there are CCL1, 2, 3, CCL 3, 4, 5, or whatever. CCL, what it stands for? The binding or the molecules, we call it ligand. Ligand. The binding molecule, we call it ligand. But the C and C is this binding ligand has special property, and usually they contain two same amino acid linked together. This amino acid is called cysteine, double cysteine. So it's the cysteine chemokine ligand, CCL. Another type of the chemokine, we also have CXCL and CX3CL, which mean cysteine, then another unknown, any amino acid, and then cysteine, or cysteine, amino acid 1, amino acid 2, amino acid 3, and then cysteine. These three types of molecules usually the common properties of the chemokines. So we call these chemokines property grouping together as CCL1, CCL2, CCL3, and so on. And also CXCL1, CXCL2, CXCL3, CX3, CL1, CX3, CL2, CX3, CL3, whatever. These are family of protein act as the chemokines. And the receptor, R, it stands for the receptor. If it's the ligands for the CCL, we call it CCR. If it is the ligands for the CXCL or CX3CL, we call it CXCR. No? And usually they are G protein coupling. The G protein coupling receptor, it means that it involves the actin myosin polymerization. So the cell, like the endothelium or the white blood cell itself, has to change its configuration, and then they can move, or it can travel transmigrated from the blood to the tissue, for example. And this is another one of the chemokine that is important in the immune system. This is just another example. We call it CCL19 and CCL21. These are specific chemokine to draw the dendritic cells from infected site to the lymph node. Help moving of the dendritic cells, mobilize dendritic cells to the lymph node. I forgot what I want to talk about this slide. Perhaps it's not so important. You can go to the figure in the textbook Abbas. I forgot about this one. But usually, just want to tell you from the cells, picture on top, you see naive T cell and activated T cell, right? Naive mean innocent T cell or the very young T cell. They are not learned what do they need to do. But once they got the in the middle picture of the lymph node, the very young naive cell, usually they circulate and then recirculate from the blood vessel to the lymph node ไปเรื่อยๆนะ on and on นะ but once this naive T cell got the signal from the antigen presenting cell, particularly the dendritic cells from the lymph node portal, the yellow the orange cell, this small pink cell together with antibody presenting cell such as dendritic cell will become mature, and become the activated T cell. And usually, surprisingly enough, this mature T cell will know where they need to go to attack the foreigner at the correct site by our circulation system. So once the T cell plus the antigen presenting cell like dendritic cells with the antigen, they become activated. And then if they are not recognized, they remain naive and they can recirculate. They are not ready to die yet. They just recirculate and they recirculate. There's small detail of the how naive T cell can recirculate, but I think maybe too much for this. But just want to let you know that there are special molecules that help to activate the function of the T cell. One is the derivative from the membrane lipid. We call it sphingosine 1-phosphate, S1P. It will help mobilize T cell from the lymph node into the blood. It means that it helps the function of the inflammation. So if you want to go research into the T cell and dendritic cell interaction, perhaps you will need to know these molecules, S1P, sphingosine 1-phosphate. And and it will get the T cell activated more easily. Oh, sorry. I think I better stop it here now, and I will try to find the class to catch you up later. Actually, it's not much, but I think I got something else to do. I'm so sorry, I cannot finish up with it. And also with the innate immunity, I should have some assignments, and probably by tomorrow I can find 1 or 2 papers for you to work out, and then on Thursday we can come back and discuss about the paper. And I communicate with you to any one of you, or Ming perhaps can help. So I better stop it here. Sorry. Any question, by the way?มี question ไหม
Don't have any question ค่ะ
เยอะไปยากไปนี่ขนาดอินโทรนะทำไมมันยากอย่างนี้วะเนี่ย
อาจารย์อ่านใจพวกหนูออกแล้วค่ะ
จ้ะตามนั้นโอเคไปละ
ขอบคุณค่ะ
สวัสดีครับ
สวัสดีครับ
Have a nice weekend
Thank you จ้ะ You too